Let's start by grounding this discussion where it really matters, at the patient level. I'm very pleased to introduce Dr. Ralph Visayo. Ralph is the Executive Vice President and Chief Medical and Scientific Officer at Vitalant. And many of you will know him as a long-standing leader in transfusion medicine and clinical research. What makes Ralph particularly important to this discussion is his deep experience in bridging clinical practice and scientific evidence, especially in understanding transfusion risk. In this talk, he's going to walk us through what we actually know about plasticizer exposure in transfusion, how that translates into clinical and toxicological risk, and where the real uncertainties still are. Ralph, I'll turn it over to you.
Okay, thank you very much for the opportunity to speak today. Let's move on and begin today's talk. So I'm going to go very rapidly. This is a lot of information in 10 minutes. Here are my disclosures, so please do hang on. Just to set the background, the medical device regulations in the EU have been tightening rules on reproductive toxin use. And in 2021, a regulation required the permanent discontinuation of DEHP use in medical devices with a sunset date of July 1st, 2030. That has been accompanied by a drop in production capacity of DEHP within the EU, and that has an impact on pricing and bag availability. And this becomes a business continuity issue for the US as well as Europe. So what is DEHP? It's a very cost-effective plasticizer for polyvinyl chloride bags. It keeps the bags flexible, prevents breakage. It is a substantial portion of the composition of the blood bag, and as a lipophilic compound, it does leach over time into the solution inside the bag, which in this case is blood. One of the good things is that it stabilizes stored red cell membranes, and we'll talk more about this later in the hour. But it is good for red cell storage, maybe not so good for some recipients. It is present in every primary container bag presently in the United States, in the tubing and the connectors. There are some red cell storage bags and plasma storage bags that don't contain DEHP, but there's enough carryover from the primary container leaching into the red cell storage bag to maintain good stored red cell membrane quality. DEHP has been gradually being eliminated from many other medical devices because of concerns over its toxicity to reproductive organs and fetal development. The chemical nature is there, and it is very quickly hydrolyzed to an active compound, MEHP, that is also toxic, and further broken down to other metabolites that are stable in the urine as a marker of acute exposure. We have nothing really to follow chronic exposure to DEHP because the half-life is so relatively short, around 6 hours. And DEHP distributes to most human tissues. In the US, about 95% of us get DEHP from food, actually. The primary intake is oral. About 75% of an ingested dose is absorbed, and the balance is inhalational because it does adsorb to soils and dust, and it can be present in house dust. It primarily leaches from food. It's not very water soluble, and frankly doesn't concentrate in animals that we eat, fish or game. So it's rapidly metabolized and doesn't go up the food chain. The other exposure is really occupational. It's very rare in places where people are exposed at the workplace in a manufacturing environment. And frankly, most of us get our greatest exposure from medical use of DEHP. The US FDA defines tolerable intake levels, and the oral dose is actually significantly less than the parenteral or IV dose. And what they do is they take animal studies looking at reproductive toxicity in animals and adjust those levels for where no or a very small amount of adverse effect is observed in these animal studies, adjusted for interspecies and interhuman variability. So the FDA defines a lowest observed adverse effect level as occurring at 0.8 milligrams per kilogram per day of IV intake. And just to give you a sense of that, the maximum dose observed in a red cell at end expiration has been 123 micrograms per ml. So for a typical red cell, we're talking about it administering 42 milligrams of DEHP in a single unit of red cells, which is in a 70 kilogram person at the no observed adverse effect level of 0.6 milligrams per kilogram. So what are the effects of DEHP? Well, firstly, in 1988, the EPA classified DEHP as probable in terms of causing cancer. That has been recently updated with more studies that in 2025 said it is not likely at all to be carcinogenic in humans. Most data on the rest of the effects of DEHP come from animal studies with rodents, rats, and mice at the highest risk because of their enzymatic systems. And in larger animals, there's either inconsistent or no effect in primate models. For example, while no studies have been performed on the effect of the bone marrow in primates very close to humans, no hematologic toxicity is seen. No cardiotoxicity has been seen in any animal. And what we also look for are effects in the cardiovascular system, whether it's hypertension, whether it's lipid abnormalities, or even obesity. And these studies have very mixed results, with some studies showing at the very highest level of DEHP and its metabolites in the urine, an association with more hypertension, lipid abnormalities, obesity, some showing less, and most showing none. Also inconsistent are studies of the immune system. While it has an adjuvant effect in small animal studies, human studies have looked at the incidence of asthma, atopic dermatitis, food allergy, have not really seen a significant signal. In some studies positive, in some studies negative, and most studies not. However, in other organ systems, there are some more plausible health effects. And while rodents are particularly sensitive to liver damage, in humans, we have not observed that. The only hepatic effect seen in humans thus far has been in occupational overdose settings where there's a mild enzyme leak. Rodents, again, also unique in that their histologic and functional kidney damage is seen in studies. But in humans, not the same thing. What we do see is a slight increase in the albumin-creatinine ratio and microalbuminuria that is inconsistently seen in humans. These are precursors of proteinuria and may in some way have to do with the animal and human studies observing impaired glucose homeostasis in people with the highest levels of urinary DEHP and metabolites, and also metabolic syndrome in these individuals with an increase in fasting blood sugar, hemoglobin A1C, and insulin levels, and even insulin resistance in the peripheral tissues has been observed. So this is a plausible effect and may explain some of the renal effects we see in human studies. But most importantly, this is a reproductive and developmental toxin. So in mature individuals, the greatest effect really is in people trying to get pregnant. So males do see a decrease in their testosterone level and sperm quality. In women, while there has not been a demonstrable decrease in fertility, assisted reproduction success seems to be a little less, although fetal losses are not observed as they are in animals. Also, there is a question of an increase in endometriosis and preterm labor. But the biggest effect really is in the developing human. In utero exposure or high exposure in the early neonatal period when a lot of blood transfusions may be necessary for very sick neonates. And what we see in male babies is a shortened anogenital distance, which is a marker of anti-androgenic activity in utero, cryptorchidism, and hypospadias. Also, in studies of both male and female babies, delayed mental and psychomotor development and hyperactivity has been associated with higher maternal levels of DEHP. In later life, central precocious puberty in these infants who become children, and as they become adults, there is a potential signal of earlier menopause and decreased thyroid function in later life. So, to summarize this, the adverse effects of DEHP are really established in animal models, but many of the concerns in humans have been disproven or low confidence finding, as we mentioned before: cardiovascular system, bone marrow, immunologic, and certainly carcinogenesis. Not really what we worry about. We worry about the adult and fetal neonatal reproductive system and other developmental toxicities because DEHP is an endocrine disrupting chemical. And the reproductive system is its primary target, with MEHP being the most anti-androgenic. It can also suppress female aromatase activity, accelerate follicular activation, which is why potentially early menopause may occur, and may have larger effects in the hypothalamic pituitary axis. As I said, there's also evidence for delayed effects in the psychomotor development and in glucose homeostasis in these individuals. So we're seeing this concern turn into the phase out of DEHP in Europe by 2030, and that will have profound implications for the U.S. blood banking industry that we're going to talk about in the rest of the hour. And with that, I will thank you and turn this over to our next speaker.
Thank you, Ralph. That really helps set the clinical foundation. Now, we're going to shift gears a bit and look under the hood at the science behind the materials themselves. I'm pleased to introduce Quentin Brabant, an R&D manager at Macopharma who works directly on the development of blood collection and storage systems. Quentin brings a very practical, technical perspective to this conversation. He's working on the exact challenges we're discussing today. He'll help us understand why plasticizers like DEHP have been so effective, what role they play in red cell storage, and why replacing them is actually a complex engineering and biological problem, not just a substitution. Quentin, over to you.
Hello everyone, my name is Quentin Brabant. I've been working for 13 years at Macopharma as an R&D engineer to develop new products and new solutions around the blood processing. So I'm very happy to be here today to talk about the mechanistic role of plasticizers in the red blood cell storage, and especially the design of non-DEHP systems. On the next slide, so you will see I've been working at Macopharma for 13 years, so of course it is my conflict of interest. Then to start, so what is a typical medical PVC container? So the first part of the container is of course PVC, and it's 70 to 90% of the total weight of the blood bag. Then we have plasticizers in order to make the PVC soft, 10 to 30% of the total weight, and we can have some additives that can be included to further enhance the properties of the PVC. So on the next slide, you can see that the main component used today, the main plasticizer, is DEHP. Why? The first part is the flexibility and the durability of DEHP. Indeed, when we are going to do the processing of a blood bag, you are going to go through centrifugation, through the press, through storage at low temperature. So it's very important to maintain the integrity and to prevent breakage of the blood bag. The second part is the processing ease. We need to have something soft in order to ensure that the handling will be easy for the operators. So that's why the DEHP makes it cost effective. And the last part is the good chemical extraction and evaporation resistance, which means that the additive solution and the anticoagulant solution will not evaporate over time, so we can reach a shelf life of 2 years, 3 years. On the next slide, you can see that we have some concerns with DEHP. The first one is that the DEHP is not covalently bound in PVC blood bags, which means that it leaches into the environment, it leaches into the anticoagulant solution and the additive solution, and finally we can find DEHP into the blood components. Even if there is a positive effect with the protection of the membrane of the red blood cells, we have also negative impact for the toxicological aspect. Indeed, we have seen long-term toxicity in animal models, and there is a risk in human, and we know that small changes in human hormone levels can cause significant development and biological effects. So it is very important today to focus on the removal of the DEHP. On the next slide, you will see that we have already some possible alternatives to DEHP. The European Pharmacopoeia has listed like the DINCH, TOTM, BTHC, and DEHT as possible alternatives. On the next slide, so it's very important to see that Today, the ban is scheduled in 2030 in Europe with the REACH regulation, but it will have also an impact in the US. Indeed, the ban in Europe will decrease the number of DEHP suppliers, so there will be a risk of the business continuity. So in the future, we'll have a hybrid market in the next years with DEHP solutions and non-DEHP solutions. We can see also more and more today regulations to ban in the US the DEHP. So it is the case in California, and we can see that we have some publications like Brown et al. To find a roadmap for the DEHP-free blood systems. Today at Macopharma, we chose the DEHT. Why? The first part is for the toxicity data that is classified as low. Indeed, the DEHT goes through complete hydrolysis in vivo. There is an inferior water solubility than DEHP, and there is an improved toxic profile with no reported mutagenic, carcinogenic, or reproductive effect. In addition to that, we have tested the leaching in the blood components, and we have seen that there is less leaching in DHT than DEHP, which is very important. So it means for the patient, there will be less DHT than DHP for the transfusion. Even if DHT is safer, we still need to find an alternative for the protective membrane effect on the red blood cells of the DEHP. The goal here is to work on the additive solution and to switch from SAGEM to PADZEM. Indeed, PADZEM has been used for decades in Germany in order to increase the shelf life from 42 days to 49 days. PADZEM has been shown to reduce the hemolysis level and to improve the red blood cell quality parameters. In addition to that, there is no impact on the clinical effectiveness and on the patient safety, meaning that we can increase and improve the shelf life of the red blood cells. We can compensate the loss of the DHP stabilizing membrane effect on the red blood cells with the PADZEM. At Macopharma, we defined a scientific pathway in order to switch from DEHP to DEHT on the safest way possible. The first step was to perform preliminary studies with Karolinska, and it was a pull and split study in order to compare DHT, PADZEM, DHP, SAGEM. We have seen that DHT PADZEM was a suitable alternative to DHP SAGEM. We wanted to confirm that, and we did a study with EFS SFS with individual blood bags. Once again, comparing DHT PADZEM and DHP SAGEM. We have confirmed the data of Karolinska with a good solution of the DHT PADZEM, and then we switched to consolidation study with a higher number of samples, n equals 30, with CBS. We have also done a bacterial proliferation testing showing that DEHT was comparable to DHP for the bacterial proliferation. Today, we start to gather data with the customer's validation process, and we did it with NHSBT to find that DHT PADZEM was suitable for NHSBT acceptance criteria. We also did the first top and bottom Buffy cut study in the US with Vitalant, showing again that we could perform DHT PADZEM top and bottom solutions in the US. In parallel, we did all the internal studies in order to ensure the quality internally of the product, and we plan to launch in the next month the real-world validation with a post-market evidence study. Today, we are going to focus on the DHT PADZEM in top and bottom for the results. So what is the top and bottom? So the top and bottom, it's a hard centrifugation from whole blood in order to have a red blood cell concentrate, a plasma, and a Buffy cut. The Buffy cut layers, it's mainly platelets with white blood cell, red blood cells, and plasma. And it's going to be further processed in order to be and to have at the end a platelet concentrate. On the next slide, you will see some results in DEHP PAGSEM, and you can see some results we have internally at Macopharma, the EFS SFS results, the NetCAD results, and the Vitalant results. So you can see that all together we have a high number of samples. And what is good to see is that we have always the same kind of results, with a hemolysis around 0.3% at day 42, with a filtration time around 30-35 minutes, and with a compliant hemoglobin, hematocrit, and residual white blood cells level, which are very consistent between the studies. Here we were focusing on the solution for the kit, but we need also to work on the process because we are going to lose the DEHP protective membrane effect. So we need to ensure that every step of the process will not increase the hemolysis. So we need to focus on the centrifugation, on the separation, the transport, the mixing, etc. You can see on the next slide that we have been working on that at Macopharma, and we have started in 2023 the best practices project. The goal was really to focus on the impact of the process in the non-DEHP solution. So we analyzed 17 key process parameters in the process that can influence the hemolysis, and we wanted to maximize the white blood cell quality parameter. At the end, with the good parameters, we can reduce the hemolysis up to 47%. So it is very important to focus also on the process. To conclude, we have seen that DEHP was very effective for the processing and the white blood cell preservation, but there are some toxicological concerns that we need to address. DEHP is a suitable alternative, and we have seen that we can preserve the white blood cell quality up to 42 days in top and bottom, and we have seen that in multiple studies. DEHP will be banned in 2030 in Europe, and it will impact the US, and it is very important today that we are working together to define the best product and the best process possible in order to have the full solution to have a safer alternative to DEHP in the future. Thank you very much.
Thanks, Quentin. Now we move from mechanisms into data. And this is really where the conversation becomes very tangible. I'm very pleased to welcome Dr. Anaïs Lotens from the Belgian Red Cross Blood Service and Nadine Marpeau from the French Blood Establishment. Together, they represent a collaboration across major European blood systems that are already actively working through this transition. They'll be presenting results from a multi-center Franco-Belgian study evaluating red blood cell and plasma quality in DEHT-based systems under real processing and storage conditions. This is particularly valuable because it gives us insight into how these systems perform beyond theory under routine practice conditions. Anaïs, Nadine, thank you both for being here. Please, go ahead.
Good afternoon. First of all, I want to thank Macopharma team for the opportunity to present you our study realized in 2023, named Quality Assessment of Red Blood Cell Concentrates and Plasma Units collected in DEHT PAGSEM or DEHT SAGEM blood bag devices. We conducted this study in France and Belgium. The study was funded by Macopharma. I will present you the study context, the aim and the study design, and my colleague will show you the results and the conclusions of the study. DEHP, for D2-ethyl-hexyl phthalate, is a common component in blood collection systems. This plasticizer will be banned in Europe in 2030, according to the REACH regulation. The replacement solution chosen by Macopharma is the use of DEHT instead of DEHP. The difference between the two molecules is the replacement of the phthalate by a terephthalate, more stable. This change of plasticizer is accompanied by a change in storage additive solution. SAGEM is replaced by PAGZEM for phosphate, adenine, glucose, guanosine, saline, mannitol. It was a collaborative and multi-parametric study. The aim of the study was the assessment of DEHP PAGZEM compared to DEHP SAGEM. We realized a storage study on red blood cell concentrates and plasma, and we followed the European requirements in terms of quality in his 21st edition. Whole blood was collected in Belgium. 16 bags were collected in DEHP PAGZEM and 16 in DEHP SAGEM. Half of each group was processed in France and the others in Belgium to finally have 4 conditions of 8 bags in the study. The blood collection bags used were top and bottom with red cell filtration. The nominal volume of whole blood collection was 475 milliliters. The separation was on Macopress. In Belgium, the separation was 4 hours after collections. The bags processed in France were transported at 4 degrees during the night and have undergone an overnight separation. Plasma were frozen between 18 and 24 hours after collection. Red blood cell concentrates were stored at 4, more or less 2 degrees during 49 days to realize the storage study. Plasma were stored at less than 25 degrees during 3 years. Red blood cells were sampled at day 1, 21, 28, 35, 42, and 49 days. At day 1, we measured hemoglobin, hematocrit, and residual cells. And during the storage study, we measured hemoglobin, hematocrit, pH, PO2, PCO2, glucose, lactate, potassium, ATP, and hemolysis. Plasma were analyzed at day 1 after 6 months, 1, 2, and 3 years. At day 1, we measured the residual cells, and during the storage period, factor VIII, fibrinogen, total proteins, thrombin-antithrombin complex, alpha-2 antiplasmin, C3a and C5a. For European requirements on red blood cell, hemoglobin must be above 40 grams per bag, hematocrit between 50 and 70%. Residual white blood cells must be less than 1 million per unit, and hemolysis at the end of storage less than 0.8% of the total red cell mass. For plasma, the acceptance criteria for residual white blood cells is less than 100 per microliter, less than 6,000 per microliter for residual red blood cells, and less than 5,000 per microliter for residual platelets. The average of factor VIII must be at least 0.70 unit per milliliter. Thank you for listening. Now my French colleague Nadine will present you the results of the study.
Good afternoon to everyone, and I would like to present to you today the results at day 1 for our study. So first of all, the first table for donor characteristics. So we have 32 donors, and we have 2 groups, PAGZEM and SAGEM, and we measure for each donor, we measure volume of the bag, the hemoglobin of the donor, the hematocrit, and we calculated the body mass index. The body mass index was calculated because sometimes we could have an impact of the hemolysis with this body mass index. And you could see here that we have similar results for all the 32 donors in each group. So 2 groups were similar. After, we have the results for the red blood cells concentrate. So for the 2 groups, we have the volume, hemoglobin, hematocrit, and white blood cells residual. And it's exactly the same results. We have no significant difference for the red blood cells concentrate at day 1 between the 2 groups. And for plasma parameter, we measure the volume, we measure the residual cells like white cells, white blood cells, red blood cells, platelets. It's exactly the same results between the 2 groups, no significant difference. And we measure too the protein, factor VIII, and fibrinogen. And it's exactly same results for 2 groups. And all red blood cells and plasma were compliant to EDQM requirements. So after, we have the storage. And the storage is very interesting for hemolysis and ETP. The units of ETP is micromole per gram hemoglobin. For the graphs on the left side for hemolysis, 1 point represents 1 red blood cells concentrate. And you can see that at day 49, 100 pags red blood cells were compliant with the EDQM requirements versus 87% SAGM red blood cells. So we can see that we have a better hemolysis with the PAGZEM solution until all over the storage. At day 1 and even at day 49, we have a significant difference between the 2 groups, and it's better for PAGZEM solution. And we could find 2 points over than 0.8% for PAGZEM for SAGM solution. And the same conclusion for the ATP. The ATP is better with PAGZEM solution, and we have the difference between the 2 groups from day 35. Day 35 we have a difference, and day 35 until day 49. So okay for that. And after we measure for red blood cells concentrate the pH and the potassium, here no big significant difference for these two parameters. Only we have a difference at day 1 for the pH between the 2 groups, but it's due to the higher buffering power of the PAGZEM. So we have nothing to say more about these results. For glucose and lactate, we have all the same results. No significant difference between the 2 groups from day 1 to day 49. Here is the plasma. So for the plasma, it's very interesting because we have the results at day 1 and at 1 year, 2 years, and 3 years. I received just yesterday the results for the 3 years, so it's very interesting for you. And for the factor VIII, we have here it's the median value because we have only 4 value in 1 year, 2 years, and 4 years. So we use the median value, and we couldn't make the significant difference, but you can see that it's a similar results between the 2 groups. And which is interesting is the factor VIII recovery at the right page. So for the median, the recovery is very, very, very good because it's more than 90% of recovery for the 2 groups and for over the 3 years. And all plasma units were compliant to EDQM requirements. For the plasma too, we have the fibrinogen and the total protein. And same as for factor VIII, we have very good results and no difference between the 2 groups of PAGZEM and SAGM all over the 1 year, 2 years, and 3 years. So in conclusion, we can see that all plasma and red blood cells were compliant to the EDQM parameters at day 1. You see that. And during the storage of plasma at 1 year, 2 years, and 3 years, the parameters factor rate, fibrinogen, and protein were compliant to the EDQM parameters, plasma. So we can say that plasma, PAXM, or SAGM are stored in the same way. The parameters controlled during red blood cell storage from day 1 to day 49 were similar for pH, glucose, lactate, and potassium. And for hemolysis and ETP, we obtained an improvement with PAXM solution. Nevertheless, all red blood cells were compliant until day 42. We can say, in conclusion, ultimate conclusion, that we have a very good collaboration for this Franco-Belgium study and EFS, SFS. And we want to thanks to all our teams in Besançon and Namur, and thanks to specific EFS laboratory in Nancy and Lyon for hemostasis and ETP analysis. Thank you very much.
Thank you. That was really helpful to see how this plays out in practice. Now we're going to bring the discussion into the U.S. context, because ultimately that's where many of us are trying to make decisions. I'm pleased to introduce Susan Lepke, who is Vice President of Government Affairs at America's Blood Centers. Susan is deeply involved in monitoring and shaping the regulatory and policy landscape affecting blood centers across the United States. She's in a unique position to help us understand what's happening at the federal and state level, how global pressures, especially from Europe, are influencing the U.S., and what this actually means operationally for blood centers. Susan, I'll turn it over to you.
Hello, everyone. My name is Susan Lepke. I'm the Vice President of Government Affairs for America's Blood Centers. And today I'll be talking about the U.S. regulatory and policy perspective on DEHP. This presentation covers the U.S. regulatory and policy perspective on the transition away from DEHP and blood collection and storage bags. The focus is squarely on what this means for blood collectors and transfusion services, not the medical device industry broadly. The global push. EU REACH drives a worldwide supply chain shift. On this slide, you can see the timeline effectively as to when everything started with this worldwide supply chain shift. The EU REACH ban, effective July 2030, is the engine behind everything else. This timeline has been pushed back multiple times, and originally the first deadline was in 2025. Nearly 40% of US medical device imports come from Europe. What European manufacturers can and can't make directly shapes what US blood centers can purchase. The January 2029 REACH authorization deadline is critical and often overlooked. Manufacturers who want to continue using DEHP in devices past July 2030 must apply for authorization by January 1st of 2029. That clock is running now, and decisions manufacturers make over the next 2 or 3 years will determine what is available on the US market. That transition is happening regardless of what US state laws say, because manufacturers are already making it work. And so many manufacturers are already beginning to meet that timeline. America's Blood Centers is working with the EBA, European Blood Alliance, and the BTA on watching what the EU is doing. I do want to note that, and this is a key insight, the EU policy does not prohibit import or use of DEHP containing products that are already banked or distributed, only their manufacture and sale going forward. The EU has stated that member organizations could still import under existing frameworks. For example, you could still sell your recovered plasma. The ban is on new manufacturing, not on existing inventory or cross-border distribution of already collected products. So where do the EPA and the FDA stand? Where are things right now? Well, as you can see from this slide, it shows the timeline of many of the decisions. FDA's history with DEHP is one of awareness without action. CDRH issued non-binding recommendations against DEHP in high-risk settings over 20 years ago. Since then, FDA has quietly removed its own DEHP safety assessment document from its website. a signal that the ground is shifting, but not a binding regulatory change. CBER added a non-DEHP draft guidance for blood collection and storage devices to its 2025 agenda, the first time FDA put this squarely on the blood and biologics regulatory radar. However, that guidance does not appear on the 2026 CBER agenda, meaning its current status is unclear. It may have been published, unpublished, deprioritized, or caught in the Trump administration's regulatory freeze. Likely, as I've said earlier, FDA is waiting to see what will happen in the EU. The critical gap is that any FDA guidance on non-DEHP materials addresses future device evaluation. It does not address the retroactive eligibility of existing DEHP cryopreserved biologics. That gap needs to be closed explicitly, and ABC will push for it when the political and regulatory moment is right. The bottom line for both agencies is they are in wait-and-see posture. There's nothing mandatory coming from either agency right now, so members do not need to respond to a federal directive because there isn't one. However, in December 2025, the EPA released a risk evaluation document on DEHP. Currently, they are speaking with many different stakeholders and partners to evaluate whether or not they will release guidance. However, they are not releasing that right now. They are currently working with stakeholders to understand what, based on their risk evaluation, what that would mean for different areas and different stakeholders. The bottom line is that the status is unclear, but both agencies are working with stakeholders and waiting to see what happens out of the EU decision. So what is going on in the US? Well, in different states, there are legislative priorities that have taken hold. Blood Centers of California spearheaded adding exemption language for blood bags in 2024, and that bill was AB 2300, which was introduced in 2024. And America's Blood Centers supported Blood Centers of California by submitting a letter to the Assembly in support of that amendment. Due to these efforts, this amendment has been included in many of the bills that you see here. The amendment was included to allow for additional time for manufacturers and for blood collectors to have compliant options while products are made available in the United States. We wanted to make sure that the blood supply was safe and wouldn't be critical. As of April 2026, we have 2 enacted laws, both in California and in North Carolina. As you can see here, Pennsylvania's bill passed the Senate on March 24th and is now in the House. And we have active bills in New York, Massachusetts, Missouri, and Vermont. The pattern so far is every enacted law has included a blood bank carve-out. California set the template, and now states have picked up that template and followed it. The precedent is holding, but it's not permanent and requires active review and maintenance. Pennsylvania is the one to watch right now as it's passed the Senate, and we are waiting for it to pass in the House. Blood was not the original focus of any of these state bills, I want to make clear. The legislative energy is coming from pediatric oncology, NICU advocates, and environmental health groups. Blood banking has been a successful add-on in terms of the exemptions. However, it can also mean that it could be removed if we're not in the room. America's Blood Centers is watching, and we are ensuring that the exemption stays in place in these bills and in any future bills to come. So the carve-out enacted in the state laws is real and meaningful. It explicitly excludes human blood collection and storage bags, apheresis kits, and cell therapy blood kits, and integral tubing from the DEHP prohibition. However, the exemption addresses what can be manufactured and sold going forward. Right now, it does not matter what happens to cord blood units that are already banked. However, we do believe and know that the exemption does cover the cord blood units because they are in blood bags. If the FDA comes out with guidance today, then members will need to work with the FDA on the banked DEHP units. Blood collectors do not need to do anything operationally right now since there isn't guidance available. The risk is real, but not imminent. The appropriate posture that we are all maintaining is to monitor and maintain readiness. Like I said earlier, FDA is likely waiting on what happens with the EU implementation, as the schedule has changed a few times already. So what is the reality of the transition? What's changed, what hasn't, and what's next? Well, what is already happening is the transition is underway. The transition happened for IV containers long time ago. Back in 2012, Kaiser Permanente announced it would not purchase IV equipment made with PVC or DEHP. Since then, many major health systems have followed, but they've been done so quietly. IV solution containers are now widely available in DEHP-free formats. And some apheresis plasma bags are already DEHP-free. However, the red blood cell storage bags is where the challenge remains, and manufacturers are already working on this solution. They are working as fast as they can, but there is not yet a regular commercially available product supply of DEHP-free red blood cell storage bags at scale. But as I said, they are working on that. The manufacturer reality is that manufacturers will have to be direct with blood collectors. They have made clear that once the transition is underway, once they do transition, they will transition fully and there is no going back. They will not be making both DEHP and non-DEHP free blood bags simultaneously. This is a complete switch, not a parallel production model. The transition is happening, and the 2030 EU timeline, as I mentioned earlier, is the forcing function. US blood collectors are operating on that supply chain timeline, whether or not any US law in specific states passes or not. So what should you do? For the blood collectors, plan for a transition, engage with your blood suppliers now, your blood bag suppliers now, and understand your time frame and ensure that when you're ready, FDA is part of the licensing pathway. What should the manufacturers do? They are going to continue to do what they've been doing, develop, validate, and achieve clearance for the DEHP-free blood bag alternatives. This requires the FDA involvement to update the BLAs and licensing for each product. It's not a simple packaging swap, and manufacturers are trying to work with FDA on this. What is the FDA's role? As you know, blood bag design requires FDA licensing, and the transition will happen once FDA is actively part of that process. As I said, they're in a wait and see pattern right now to see what the EU is doing, but they are available and willing to work with blood collectors and manufacturers on this issue. There is no immediate requirement to take immediate operational action right now because no federal mandate exists. However, ensure to be ready when that mandate does drop. So at America's Blood Centers, we are actively monitoring the situation. Our first priority is to protect the blood bank exemption in every state bill. The exemption really is to ensure that there is a safe and available blood supply and that blood collectors can continue to supply blood using the bags that are available. This is where blood collectors' engagement matters most. California's exemption language is the template and is the standard and is included in the bills that are already going through the legislative process. However, we need your help in terms of any new bills that come out either this session or next session to make sure that the exemption language is included. Our second priority is to monitor both the FDA and the EPA, but not engage early. The risk of overengagement, if we push too hard for FDA guidance before FDA is ready to move constructively, we risk getting guidance that is poorly written or creates more problems that it solves. Timing matters. And also, the FDA doesn't want to publish guidance or information in case the EU decides to change their timeline again. Additionally, the EPA is looking and working with stakeholders to ensure that whatever guidance that they do create does not disrupt the process, the medical process for other organizations and stakeholders. So what does ready mean? It means that ABC will be prepared so that when FDA and the EPA does move, we can engage immediately and credibly. We are currently ensuring that we have the lines of communication open and are ready to move as quickly as those agencies are ready to move. We are also watching for any shift, making sure that we are part of the conversation, and also on any EPA action and making sure we're communicating with the EPA as well on DEHP classification or any healthcare exemption. Also, we are watching the administration to see if they will also push for any action or changes that may impact the FDA or EPA's regulatory stance. So the key takeaways are that the transition from DEHP in blood collection is not a question of if, it is a question of when and how. The EU 2030 timeline is the forcing function, and manufacturers are responding to it. For blood collectors, the most important near-time action is to stay engaged with your blood bag suppliers and make sure you understand their transition timelines. At the federal level, both the FDA and EPA are in a wait-and-see pattern. There is nothing to respond to right now, but both agencies are trying to collect as much information as possible. ABC is watching, and we are working with the agencies. At the state level, both the active legislations are moving, especially in Pennsylvania. But if you have members or staff in any of the states that have active legislation, this is the moment to engage to ensure that the blood bank carve-out stays in there. It's not guaranteed, but our job is to keep it there to ensure that we have a safe and available blood supply. America's Blood Center is positioned to protect the blood banking carve-out, engage manufacturers and the FDA and EPA on the transition timeline, and ensure our blood collectors and those in our blood industry do not get caught off guard. So I want to thank you today for my presentation. My email is on the slide. If you have any questions, feel free to contact my team and I, and we will work with you to ensure that the DEHP transition moves calmly. Thank you so much, and I will now turn it over to Dr. Cohen for questions.